I'm Timo Dickscheid. I think most of you know me, one of the people working in the BigBrain project, and I will tell you a little bit about some tools that we developed that allow to work with atlases and high resolution data, such as the BigBrain. And I will show you a few things you can do with that, and we will also do some hands-on exercises where you basically can go along with things that I'm showing. All right, so I will tell you something about our Zebra tool suite and particularly focus also on how this can be used to work with the BigBrain. What are we doing? We are mostly dealing in our research group with image analysis. So we develop methods and tools to enable high throughput microscopy. And of course, one of the things we are doing is helping to develop BigBrain and the related data sets, models, and softwares. So we deal a lot with data management, a lot with AI-based image analysis, and also as a third pillar, developing software to work with high-resolution image data and brain atlases. And the thing on the right is what I will be mostly talking about here today and check out with you guys. So I'm not sure if you have seen such things, maybe in the talk by Nicola, but the data that we are most interested in is mostly coming from histological imaging. So we are cutting donor brains in thin slices and then imaging them under microscopes with different modalities. The most common one and classical one is the one that you see here on the top right, where the sections are stained for cell bodies and imaged with a light microscope. In that case, we usually have images with a resolution around 1 micrometer. And the BigBrain is made up of such data, only at that time at a reduced resolution of 20 micrometer. We also have different data. The one on the bottom is particularly nice to look at. It shows nerve fiber distributions that we can acquire with polarized light imaging. And there are other modalities, but in this in this tutorial today, we will mostly deal with such cell body stained data. In the last years, here on the European side, we have been working a lot in the Human Brain Project and building up infrastructure, research infrastructure for neuroscience that is called EBRAINS, a European infrastructure for brain research. And I was responsible with some other people to develop infrastructure for brain atlases. So currently EBRAINS is accessible online. There's a link below, and you can find their atlases of the human brain, of the rodent brain, and also of the monkey brain. Actually, for the monkey brain, Nicola Palomero Galapa, who talked here yesterday, is doing a lot of work. And what the tools that we will be showing, they actually support these atlas services in EBRAINS, but they can also be used outside of EBRAINS for the most part. If you go to the website ebrains.eu, you'll actually find different categories of software and infrastructure services for data sharing, for brain simulation, also for health research. And the one that we are interested in here is the category for brain atlases. That's where you will find information about these atlases and also entry points to the tools we are discussing today, although you typically also just have a simple URL or GitHub link to access those tools, which we will be using today. The tools comprise software for accessing and working with brain atlases, tools for integrating new data to brain atlases, and tools for analyzing data with the help of brain atlases. What we discussed today is mostly the left side and a little bit the right side. I will not discuss so much how to integrate new data with brain atlases, although that is of course an important topic. And so if you have specific questions about that, happy to discuss them, but I haven't planned to talk a lot about this part. What I will focus on is of course mostly the human brain and the human brain atlas that we have been building in eBrains. And I would like to start by explaining briefly the concept of this atlas, which we call a multi-level atlas. The idea is here that we connect information from different spatial scales and also from different resolutions and make them really usable together in a coherent framework. And of course, the most common perception of a brain atlas is that of a labeled brain volume at the millimeter resolution, such as this one. This is the maximum probability map from our cytoarchitectonic atlas, the Jülich brain atlas that we develop here on site in Jülich. And this is a labeled brain parcellation map, which basically means each voxel, or in that case, each vertex on the surface, is assigned one deterministic label, which indicates which brain region has been assigned to that particular position in the brain. But the cytoarchitectonic atlas that we developed, and also some other atlases, have a probabilistic nature. So behind these regions, we actually have measured how the shape, the morphology of each region varies in different individual subjects. In this case here, this is based on delineations between different cytoarchitectonic regions in different postmortem brains. And these have been aggregated into the MNI reference space at the millimeter resolution. And by warping those postmortem brain delineations into the reference space, of course, we get contradictions because of the variability in the different brains. So the delineations will not coincide in the reference space, and therefore we get such a distribution over the brain, which indicates the uncertainty and the variance of each brain region shape in the space. I will get to this in a bit more detail later on. So this somehow captures already the variability in different individuals. But this is then still information aggregated at the microscopic scale at the millimeter level. But of course, the delineations are based, if they are based on cytoarchitecture, they stem from much higher resolution primary data from these histological sections that I've shown to you. So behind such probabilistic maps, we always have histological sections at much higher resolution and precise delineations in the background. And one important idea of the framework that I'm presenting is to connect this, so to actually make these high resolution image data and measurements accessible by going from the lower resolution reference space. So we want to build a link, as it's shown here, from the millimeter or centimeter even to the micrometer level. And actually then being able to zoom in to the level of detail where we can see individual cell bodies. And this is exactly, so this picture here is exactly what we will go through today, later on in the tutorial, with our hands on. So you can do more or less exactly what we see here by using only a web browser or by using a Python library that we will show you today. This, so to say, walk across the spatial scales is just within one modality. So the regions in the reference space, as well as the detailed image data, they refer to cytoarchitecture, so to the distribution of cells in the brain. But of course, there are other principles of brain organization, which can give rise to other schemes of parcellating the brain and to other measurements. And so in the framework that I will be showing, we also try to include other modalities. Here's only a few examples. That will be other parcellations, but also distributions of different features in the brain, such as neurotransmitter receptors. I'm pretty sure Nicola yesterday talked about that as well, or connectivity, for example, connectivity matrices from neuroimaging cohorts, which have been linked to brain parcellations. So many different things linked into this framework. And the challenge is, of course, how to make that accessible and how to enable to navigate such a complex system of interlinked data sets of different types and sizes and resolutions. For this, we have developed a web viewer, which allows to zoom in to very high resolutions and in 3D. The one that I show you is called siibra-explorer. Zebra is written with 2 I, and it stands for software interfaces for interacting with brain atlases. And there is also a Python library that's called siibra-python, that can do the same things, in fact, shares code with the online web viewer. So we have two entry points, one for interactive use and another one for programming. And of course, we connect to this concept also analysis tools, which I will not go into too much detail, and spatial anchoring tools. As I already said, this will not be in the focus of today's tutorial. So the focus will be the Zebra tool suite, and it has mostly three components. That's the siibra-explorer, the online web viewer, we will look at that soon, the Python library, and there's also a REST API, an HTTP API of Zebra, which we actually use to expose functionalities from the Python client to the online explorer, but it is documented and could be used for application development. The focus today will be on the left two, the Explorer and Python. So before we dive into this, just a brief mentioning, the Python library is now at version 1, but we are still heavily developing it and including new functionalities. And you will, as we go along, you will see many different data sets in the brain, and you will probably often ask the question, how many of those are there, or is there such a data set at each location in the brain or not? I will comment on that, but in general, it is important that you know the data sets that you see here are coming from an online repository, and the amount of data is constantly growing that we have linked here with the ecosystem. So I will show you some data, and a lot more is already there, and a lot more is still in development and in the pipelines by different labs who work with us. So it's really growing in the background. And then the last message, if you see anything here in my talk where you say, well, that's not intuitive, or I never expected this to work the way you show it, or if you want to interface with some of these tools because you develop your own tool or use it, please just raise your hand or use the chat, and I'm happy to discuss, or even contact me after this tutorial. Okay, I will go through some of the tools now, show you some things you can do with it, and you can just do this in your laptop if you have an online connection as we go along. First thing I would like to show you is how we link data features to brain regions. So that's the part of the picture that you see here, which I try to show you now in an online viewer. Just before we launch it, I would like to give you an overview of the basic concepts behind that, so you understand it. In the Zebra tool suite, the main pattern that we have implemented is basically that you go from locations in the brain to measurements and data elements. And locations in the brain can be very different things. Yeah? It can be brain areas from a parcellation, such as a cytoarchitectonic brain region from our atlas that I just shown. But it could also be unnamed brain structures, so coordinates that you have clicked in a particular reference space, let's say the MNI space, or a bounding box, or even a structure depicted in an image. So for example, if you think of an fMRI activation map, which has a certain activation peak or blob, that is also a location in the brain if you bring it into a reference space, which you can use with Zebra. And actually there is a link between such named brain regions coming from brain parcellation and, let's say, unnamed user-defined structures in the brain. A twofold link. One is, of course, we can access the maps of the brain regions in a particular reference space to actually end up with an image structure as well. And we can do the other way around. We can take points or structures or bounding boxes and assign them to brain areas. And this is also functionality that you find within Zebra. So you can actually go forth and back between these locations in the brain. What Zebra then offers is a way to query information, to query especially different data sets. And the unnamed locations in the brain, they can be used to run spatial queries, so to find data that is linked to coordinates in reference spaces. And once you have a brain region from a parcellation, you can also find data sets that have a semantic link to the brain regions. Yeah? So there could be data sets that have been published and have been formally or semantically tagged by a certain brain region. Both of this can be done, and it can also be combined through the links I have just shown here. When you run these queries, you get data features in Zebra, which is more or less a description of a data set that's there. Think of it as metadata. Yeah? It tells you that there is a data feature that has this link to the brain and that has a publication somewhere, an online resource, and so on, mostly metadata. And then usually there is a step that we call fetching, which allows you to actually get the data behind such a data feature. And in this last step that you see here on the bottom, you typically leave the Zebra world and get your hands on interoperable data structures. So what we have here on the bottom will typically be, if we think in Python for the moment, not in the online viewer, this will typically be a pandas data frame or a nifty image. It will be typically a structure that the tools that you work with know are compatible with, not a Zebra structure. While everything above are structures in Zebra, and they have much more knowledge under the hood. They typically know about the reference space, about the brain parcellation that they are coming from. Yeah, so the fetching mechanism at the bottom usually brings you into the world of interoperable data sets. Everything else is in the Zebra framework. When we link data sets to locations in the brain, I explained it already briefly, there is this possibility of semantic tagging. That is then usually just the name of the brain region, yeah? or an identifier of the brain region. So for example, we can have a receptor density fingerprint, which has been linked specifically to the primary visual cortex, maybe even particularly in one hemisphere. So this is not linked to a coordinate per se, but to that name. Then you can have spatial primitives, coordinates, and so on that are linked to a particular coordinate. And you can have image data that has been registered to a template space. You know, these are different ways of linking data to the brain and to the atlases. And these links can be used in the tools that we are looking at now. So long story, long introduction. Let's now go through the viewer and look at a few of the things that I've shown you. The link is already in the chat. I'm switching over to my browser. So atlases.ebrains.eu/viewer. And then there we select the human atlas. And what we see here is already the maximum probability map of the Jülich brain cytoorganic atlas in the famous M&I space coming, of course, from the Montreal side. So this is a typical 3D viewer as you would expect it, except as people in Montreal keep reminding me, you cannot move around the crosshair, but you move around the image. This has technical reasons. We are still discussing this, but otherwise it's just a 3D viewer. Yeah? So you can move around the brain. In the bottom right, you have here a 3D view which you can use. You can use the scroll wheel to zoom in, and of course in the M&I space, zooming in at a certain point does not reveal more information since we are here operating at a millimeter scale. You can even select oblique cutting angles. So if you hold the Shift button on your keyboard and then click somewhere with the mouse and draw and pull, is this the right word, pull? Drag, maybe drag. You will actually select different cutting angles as you see here, right? This is of course something that you usually not do a lot in such macroscopic resolution views, but we will see later that it's very useful if you go to higher resolutions. You can always restore the orientation or the position here at the top. Here's a location bar. When you click there, you can restore this to the standard zoom, the standard position, and so on. So here we are back. You have to slow me down if I'm talking too fast. Yeah, I have seen these things so often. I never know exactly hard or easy it is for you to follow along. In this atlas, you can select a brain region. Let's here select the primary visual cortex just by clicking on it. And what you see here is that the side panel opens for that particular brain region, and it has automatically loaded the probabilistic map of this particular brain region. Yeah? This makes only sense when you select the brain region because the probabilistic maps of the different regions overlap. So it would make sense to show them all at a time, right? That is why we only show them when you select a particular region. Here you see the probabilistic map. And the side panel gives you information about the selected brain region and the map that you have selected. So you see here, of course, the name. You see to which atlas it belongs. You see where the centroid is. If you click on that, it navigates there. And you see here a DOI for that. I will get to this later. A DOI for this region, I will get to this later. A description as well. And you will also see that there is a second panel here in the sidebar, features. If you click on that, you will actually see, get access to the linked data features. There are thousands at the cellular level. This is something that will change in the next version that we are releasing soon. So if you open that here, you will see that actually there are intensity profiles from the BigBrain, which fall into the selected brain region, and there are of course many of them. In the next version, those will be grouped together. But if you click here on filter, you see that there are different types of cellular features. For example, we also see here, if I disable that, layer-wise cell densities. There's only one of those. If I click it, you see here description of this data set, and you see also visualization. And what you see here, I'm sorry, I have to move around my the control elements of the Zoom call. Just a second. How do I move that away? Okay, so what you see here is actually average densities of segmented cell bodies in the primary visual cortex and their standard deviations. Yeah. And this is one of the data sets linked to the selected brain region. You could also, in the features, select a different group, for example, molecular, and here you will find receptor density profiles, which, as I said, I think Nicola might have talked about these. So you can also select the receptor density profile, also has a DOI, and it also has a visualization. So you see here such a profile, you can also download the data here. Going back, I just close the side panel, which brings me back to the atlas itself. You see here on the bottom of the viewer, there is a chip where you can select different brain reference templates, and we are currently in the ICBM asymmetric template. You see here a little information icon. This shows us information about the currently selected brain reference space. And there is a button detail, which shows us, will bring more information. I will get there soon. There is also a chip for selecting a brain parcellation. Currently, we have selected a cytoarchitectonic parcellation in the newest version, but there are also others. For example, here are also parcellations of fiber bundles, for example, which are also probabilistic. Yeah? So if I select such a fiber bundle here with a single click again, it will also show a probability map. This comes from Paris, from the lab of Jean-François Magin. And here there's also an information button which shows you information about the selected parcellation. If we go briefly back to the slides, you see here that the idea in Zebra is that we separate the actual management of the data set from the interaction in the atlas. What does that mean? All the things you see in the atlas, the brain parcellation maps, the reference templates, the data sets that have been linked to brain regions, these are, with very little exceptions, these are all data sets with a DOI that are curated and most of them are available in the data sharing services of EBRAINS. There is a metadata database, a graph database that's called the EBRAINS Knowledge Graph. So you could also find those data sets by searching there most of the time. The Atlas really uses these curated data sets and brings them into the anatomical context. And one key idea is that you can always find the original locations of these data sets. And this is what I already indicated. So if we just go back where we were, we select one probability map here. There's this DOI, and if you click on the DOI, it will actually take you to the original place of the corresponding data set. So you can see here that this probabilistic map of the primary visual cortex in the cytoarchitectonic atlas is itself a data set. So not only is the atlas a data set, each of the probability maps is a data set. So you find here detailed information about this data set, you find publications, you find also from here the links to the viewer. We don't need that, we already came from the viewer. You can here download the data in different forms. As I said, you find the corresponding publications. You find information about the specimen. As I explained earlier, each probability map comes from mapping the brain region in different postmortem brains, and you can actually see these postmortem brains here. Of course, you cannot find this is anonymized, so these postmortem brains only have numbers, but there is information, and typically you can even for these brains find some metadata that is publicly available, for example, the age and the gender. And this is not only the case for the brain parcellations. Yes. So if I again here go to the related data sets, let's say I go to one of those receptor density data sets again, there's also DOI. If I click on this DOI, I will find the data set for these receptor densities. Yeah. See that here, different authors, different publications, typically also different specimen that were used. You can see that here they have a different ID that was not measured in the same brain. You can find all that information here in the knowledge graph in the background. The same holds also for the reference templates. So for example, we saw here that the current reference template is the ICBM, nonlinear one. If you click on detail here, this will not bring you to EBRAINS. It will bring you to the server at McGill, which is hosting these reference templates, yeah? Because that's where we got them from. As I said, not everything is in EBRAINS. Most of them are in EBRAINS, but not all of them. Yeah. So that is one key idea, to connect data from different resources across modalities and scales into an anatomical reference space with the help of atlases. I think we did this. Since we have only part of the people on site and many online, please let me know if you are following along and have questions or have trouble. So it would be now a good time. If you do that and you're stuck anywhere, just let me know. Is there something in chat already?
It's behind you, so I observe it.
Okay. Then let's move on from here. So we basically had a look at the macroscopic scale and how different data sets are linked in the online viewer. We stay with the online viewer for the moment, and I would like to show you how you can actually get from here to high resolution information, to the microscopic level, down to individual cells. So we're doing this from the same place, still in the viewer, just by switching over to the BigBrain reference template, which you should have heard about already in this course, probably about the BigBrain. Is it? Yeah, Susanna?
Brian showed something yesterday.
So the BigBrain is a 3D reconstruction of a postmodern human brain that we published with McGill and Jülich together in 2013. So we basically took all the histological sections, more than 7,000 of this particular brain, reconstructed them back into 3D at a resolution of 20 micrometer and made that a 3D model. This data set is 1 terabyte of image data, and this is usually hard to work with. Yeah, it's because it's so huge. Still 10 years or so after the publication, dealing with a 1 terabyte image is not an easy thing to do on your laptop. Still not today. We can go to the microscopic scale just by switching the template. Before we do that, I want to briefly mention when we change the reference template, we are changing the coordinate space. Yeah. So when we go from the M&I space to the BigBrain space, that's a different coordinate space. The BigBrain is in this originalist logical reconstruction asymmetric. It has a higher resolution, and it's so we are changing the coordinate space. And the question is, how do you link these coordinate spaces? Right? Previously, we have linked data to brain regions. Now the question is, if we switch the coordinate space, the template, how do we connect those? And there are, of course, several ways to do so. One way is if you have in the different template spaces the same brain regions mapped, then you already, of course, have a link by finding the same brain regions. Right? There are parcellations that are based on macro anatomy that you can always apply, like the Desikan-Kiliani to a brain and orient yourself. For cytoarchitecture, it's not that easy, but we have mapped cytoarchitectonic brain regions in the BigBrain. They are not yet complete at full detail, and of course you find them in the M&I space. Yeah? So in some parcellations you find the same brain regions mapped in different templates, so that gives you a link between these different reference templates. And of course, a second way of connecting them are spatial transformations. So you can compute a registration between the two brains, and then you basically know from the registration result which voxel in the one reference space belongs to a voxel in the other. And the viewer has both of these concepts implemented and is using them in the background. So as we switch from the M&I space now to the BigBrain, it will actually try to preserve the current view. Yeah? And that's a very powerful mechanism. So what this allows us is, for example, here in the M&I space to select a brain region. Let's say we select here area 4P that is in the motor cortex. Move there just as we have explained above, and then I zoom in, and of course there is very limited resolution here. But what I can do now is I can here go to the chip on the bottom, which has the reference templates, and select the BigBrain. And the viewer will then switch to the BigBrain, but also try to stay close to the zoomed in region, right? So you see here that the area 4P is in the online version of BigBrain, there is no map available, so we don't see the brain region map here for 4P in BigBrain. But since we were in the center of the probabilistic map, and we are now at the corresponding voxel in the BigBrain at the same zoom level, it is very likely that the position where we are here is also in the same, of course it will be in the same hemisphere, but it will very likely be in the same brain region, right? And maybe, for example, Olga, who is here, might be able to confirm this. I'm not so good with this. For the motor cortex, there are actually ways to verify it. We will look at that soon. So now we are here in the BigBrain space. We have much more a level of detail. We can zoom in, again with a scroll wheel, or on my Mac, I do it with my 2 fingers. And as I zoom in here, you can see that we see larger cell bodies as black dots, but we don't really see the morphology of the individual cells and so on. We can get an idea of cortical layers right here, but we don't see the individual cells. And of course, in the BigBrain projects, we are all working to increase resolution, and at some point there will certainly be a brain with higher resolution available. But we have already increased the level of detail here by connecting image data, partial image data with higher resolution. And you can actually see here on the top left, there's a little 10, which tells me there are actually 10 data sets linked here to the current view. So if I click here, sorry, that was It says that there are 10 spatial features linked, and if I click on that, it actually shows me where they are. So you see, obviously here there are in the current zoomed in view, there are some sections, some brain sections in the coronal plane. In fact, they refer to the original BigBrain sections, which have been anchored to the BigBrain dataset and have been integrated here with the system. You can actually click on one of those boxes to load one, or you can also select them here, just as we did it previously with the regional features. We can also select them here from the system. So I do that here. I select the histological section number 3601 in that case, and if I click it, the viewer will move to that plane, which was very close by, of course. And you see, again, here's some information about this. Here is a DOI. Yeah, so I can click on the DOI, and you will see that these 1 micron sections are also a data set with descriptions online. And what you see here, if I close this for a second, now I can zoom in, and in the coronal plane, I have now the high resolution of this section, which has been scanned at 1 micrometer level. And this is probably also very helpful to verify whether we are here somewhere in the motor cortex region. We might be looking for larger cell bodies there. And to me, as a computer scientist, it looks very much like we have here some really big ones around. So maybe we are indeed here close in the motor region. Yeah, so you really have here the full detail. What you also see is that in the other sections, in the orthogonal sections, of course, there is no higher resolution, since this is a 1 micron scan of a histological section that was cut at 20 micrometer. So when we look at it from the side, it still doesn't have a higher resolution, right? But you see the big difference. And if you want to work and explore such a section, there's a nice feature in the viewer, which you see here in each of these planar views. There is this little icon that allows you to maximize each planar view. So I use that here to maximize the coronal plane, and then I basically have a 2D view logged in. I still see on the bottom where it is in the brain. Actually, PJ, if you're still online, did you see that we modified this? Because we had a nice request here on the bottom. This has meanwhile been implemented, I think. So we can explore here this 1 micron section. And as you see, as we go from the left to the right hemisphere, this overview here is also switching the view. That was exactly what PJ reminded us on in one of the last workshops, and it's now working. So you can explore the 1 micron section. As I said, in the BigBrain, it is really possible to see the layering structures in the brain, right? So if we go here, we very clearly see layer structures in the cortex. And many of you might know that there is also a map of cortical layers available for the BigBrain, which has been developed by Konrad Wachsbühl and some of the people here as well. And this is, of course, available here. So we can select from the parcellations here the cortical layer maps. You will not find them in the MNI space. Yeah, they are only available here in the BigBrain space. If we select them, you see here nicely the classification of the cortical ribbon into the 6 different layers. And you can also click again the information icon here, and you see information about these maps. Again, you find a detailed link to the underlying publication, and you can see here that this is comes from this work of Konrad Wachsbühl and some of us. Closing this here, and this is particularly interesting to see in conjunction with such 1 micron sections. Yeah, so if we zoom in here again, and we choose once more the 1 micron section. A bit unintuitive here, I understand this. So I select the same one again for convenience. So now, if I look at our micron section again and I hover with the mouse, you can actually see where the layers have been mapped here. You see that they are, at some places, they are really pretty nicely shaping up, even here at the cellular resolution. Some places it's not, so you see here, for example, here's a histological artifact, but the layer is, since it follows a certain smoothing and data model, it extends here into the other place, but it's pretty nice, and you could actually here see how the large neurons, the large cell bodies actually sit in layer 5, where we would also expect them. I told you about, by the way, when you are in a maximized view of one plane, if you hit space, you cycle through the different views. That is also quite useful usually. So if you want to have a quick look where you are in the sagittal and axial planes, this is just the hit of the space button. And here in the 3D overview, you can. It's very useful to actually get a orientation in a macroscopic view where you are. What you might also notice is that when you're here in the 3D view, when you zoom in, you don't get the high resolution data, right? So this 3D view is really just a surface mesh, and the lower resolution version of the image is thought to give you a 3D orientation in the brain. This view is not thought to give you the full detail. Yeah? So for the full detail, you usually go in one of these orthogonal sections. Let me just close this here. And what you should also notice again is this possibility to do the oblique sections. So while in the millimeter space that was not super useful, it is now really quite interesting if you zoom into a certain location in the brain, maybe height, parcellation again, and then again if I hold the shift button and move along, I can really here see the brain from at different cutting angles, and that is actually quite helpful in the BigBrain, because if you want to study the layering structure of the cortex, depending on where you are, the native planes are not nicely aligned with the cortical surface, right? So you might, the native X, Y, or Z plane might be very oblique relative to the cortical surface, and then you can, it's probably here already, let's choose another example. For example, here, I think this is a quite oblique place here, so we don't see cortical layering here. But if I then use this oblique sectioning, I can nicely try to find an angle where I see the cortical layer structure at this place, right? And then again, you can always go back to the native resolution and angles here with these buttons at the top. Yeah. One thing that I should mention is this online viewer encodes most of the state in the URL. What does that mean? If you are somewhere like here and you copy the full URL and you send that to a colleague, they will see the same thing you see here. This applies to many of the features, so selected brain regions and so on, try it out. This is a very easy way to share something that you see, or even bookmark it for yourself, right? So you could just, if you zoom somewhere and you see, wow, this is such a beautiful cell or something, you could just bookmark, yeah? So for example, this black dot, I could just bookmark it. You can try that out actually. I just copied the URL and put it in the chat. And if you click the link in the chat, you should, I didn't test that to be honest, I'm a bit scared now, but actually you should see the same black dot if you hit the URL.
You do, but you see the layer structure.
Yeah, you see the layer structure because I have just hidden it.
Oh, I see.
This, you can hide this here when you click on the eye.
Yeah.
Yes, we see.
Yeah. If you want to make more use of that, the viewer also allows you here on the top to click to copy the positions, and you can also create a short link here. Creating a short link that is permanent will require you to log in with your org ID or EBRAINS account, but you can also do it without logging in. It's just that then I think we will not. You cannot rely on the URLs to stay forever. But you can really just copy the URL. This should work. Before we will soon also look into Python, but let me just for a moment show you a few more things here. So one thing that many users of our software have not discovered, although it is pretty useful, is that you can also do annotations here. Simple ones, so there are of course much more sophisticated annotation tools around, but when you go here, when you look at the top right, you don't see my mouse when I'm going there, but on the top right there are actually also some icons. There's a help icon, there's a download button, and there is this grid icon here, and behind the grid icon there you can find some plugins. And one notable plugin is this annotation mode. So if you click on the annotation mode, a side panel will again pop up, which gives you the possibility, for example, here to select such a polygon tool. And I can use that, let's say, I'm just doing it here, doesn't matter, to annotate a structure. Yeah? As I said, this is really a pretty simple annotation mode. But I have now entered a polygon here. You see that on the left. I could give that polygon a name, yeah, my nice oblique cortical patch, whatever that is. You see that it has four vertices. I can move around these vertices, and I can download and share this annotation. It will also in your browser session. It will stay there. If you log in, you can do a few more things. Here's an old one that I did, which I just delete now. If you click here on the export button, you can download it, and you see that it is represented by a JSON definition. Yeah, that's a bit lengthy, but I could actually just copy that here, and I will show you in a few minutes why that is particularly useful. Yeah? Of course, it's not very readable, but I can also click copy to clipboard, and then I have the annotation in my clipboard. But as I said, it stays here. For example, if I move away in the brain, I move somewhere else, I could still go back to the annotation mode, open it here, click on some of the vertices, and then I'm back there in my annotation. Yeah. Okay, I'm closing the annotation mode now. The annotation will still be there.
Timo, just a quick question from our side.
Yes.
The annotations, are they 2D or 3D?
Yes, they are 2D. It's a very simple annotation mode.
Okay.
So that is far from what you can do, for example, with Atelier 3D. It's really 2D, so you annotate in the section. You could, it's not exactly 2D. It's the most complex thing that it can currently do are polylines in 3D. So you could put a line with vertices in different planes. Yeah, that works, but you could not annotate a surface or something like that. Yeah, but you could very well have a non-planar polygon. That works. Yeah, but it is really very basic. So if people are interested in that, it's we might revisit this annotation mode in the future with some more sophisticated features. At the moment, it's rather simple. It started with the idea just to annotate individual points and give them names, and now it simply can do a few more things. Yeah. So you would at the moment, for example, use it to annotate a cortical profile that you're interested in or things like that. Not really for brain mapping. All right, I'm switching back to my slides for a moment. So we did this, yeah. We switched to the BigBrain, we found some anchor data, and I would now like to show you a few things in Python. And for that, there is this link. I think we had it already in the chat. If you click that link, it will take you to a normal GitHub page, and we maintain here a set of Python notebooks with some tutorial material. If you scroll down here, you see on the right there are buttons where you can launch the notebooks either on EBRAINS, which is hosting a Jupyter lab, or on MyBinder, which is another cloud service where you can run Python notebooks. What I would suggest is that you start one of them already, and maybe the one that is called cortical patch sampling without going into it, just because it takes a little bit of time to load it. So when you click here on the one with the number 6, either on launch binder or lab.ebrains.eu, you will run it. For lab.ebrains.eu, you will need an eBrains account. It's very easy to get one. You just go to ebrains.eu/register, but if you don't have one right now, I suggest you use the launch binder button instead. So I will do that here as well. We will look at that notebook later on only. Oh, I already loaded it earlier on. Just launch it so that it loads. In the meantime, I would just show you some basics by going at the documentation, siibra-python.readthedocs.io. The link is also in the chat. So with the Python library, you can do everything we did in the viewer, really everything in a programmatic fashion and more. It can do a lot of things that we will not be able to cover all now in this 1 hour that is left, but I will show you some key things. What is very important is that here on this Read the Docs page, there is a step-by-step guide, which actually contains working code snippets for quite a few things it can do. So you can read through the documentation anyway. It is somewhat complete, but the easiest way is really to look up some code snippets here in the step-by-step guide. Let me just click a few, and there you see the executed code online without running it. So if I click here on atlases and brain parcellations, for example, you find here a notebook. At the bottom, you can download it, which basically shows you how that works. So Zebra, you really, as you will know from Python, you just import the package, and then you, maybe the first one is not a good one because that's not something we did. Let's take the second one. So you find all the elements that we saw in the viewer here in Python. For example, it has a registry of pre-defined parcellations. And you can simply fetch such a parcellation or select such a parcellation by providing a keyword. You can also use tab completion, but usually we put keywords. For example, you say parcellation.get Jülich, and it will give you an object that represents the latest version of the Jülich brain cytoarchitectonic atlas. In the Python client, there's one very important pattern that we have implemented. You will usually load data only very late when you really need it. So at the moment where we get such a parcellation, we don't load a lot of data yet. We just load the parcellation as such, which is merely the name of the parcellation, the link to the data set, the names of the regions, and so on. At that stage, we have not loaded any parcellation map or something. We just have an object that represents the parcellation more or less in terms of names. And you see it shows you some URLs and so on. We can choose other ones and we can find some metadata. Whenever you want to access a parcellation map, there will be an additional step included. And this is here in the second section of the step-by-step guide, maps and templates. So I'm choosing here the third one, which is about accessing parcellation maps. So here you will see the same thing. We show the available parcellations, but here we actually say get map. And getting a map requires us not only to specify the parcellation, but also the reference space that we are interested in. Yeah? So when we were looking in the viewer, we saw that many maps have many parcellations are actually mapped in different reference spaces. So when I want to have a map, I'm specifying also the space in which I would like to have the map. So here we say get map. We want to have the Jülich brain parcellation, but we want to have it in the ICBM space, which is here just identified by keywords. You can also say M and I, it will also work. And we specify a map type, which is here denoted labeled. This is necessary to distinguish the probabilistic maps from the labeled maps, which we also saw in the viewer, right? So if you would put here statistical, we would actually get access to the probabilistic maps. If you put labeled, we get access, in that case, to the maximum probability map. And then for these maps, this still did not load the image data. This gives us an object that represents the map. It knows about the map, it knows about the map brain region, it knows about the colors in the map, and it knows from where it can get the image data. It still did not load the image data. As I said, we always follow here a lazy scheme of loading data. So it will only load data when we need it. Otherwise, when you load the Zebra library, it would take you 15 minutes to load everything it can actually provide. That wouldn't make sense. So when we really want to have the 3D map, we have to fetch. And this is really an explicit call here called fetch. It can have arguments, but in the simple case, you just say fetch, and then you actually get the image of a map, and that is a nifty image. Yeah. Going back very briefly, if you remember this overview that I showed you, this is exactly the bottom here. Yeah, we have a Zebra object that actually defines a data set. It defines a certain concept, but we need to do an explicit fetch step to get the underlying data. And the underlying data then comes in an interoperable format, which is in that case a nifty image. Yeah. So as we call fetch here, we really get a nifty image. And this nifty image, we can just work with it in Python as you would when you load it from disk. For example, in Zebra, we typically use the nylearn plotting module to display the data. We didn't re-implement this in Zebra. This works very nicely, and as soon as you have a nifty image, you can use that right away. Here in the same section of example code for maps and templates, there's also an example for BigBrain, and now comes one of the interesting parts. Because if you want to work with BigBrain, it really works the exact same way, although there is a very different data set, right? The BigBrain is not available as a nifty file usually somewhere because it is 1 terabyte in size. But you still work with it in the very same way. So the BigBrain is not a parcellation map, it's a template. So to get it, we would... I just increased the font size here a bit. We would basically say get template. I will show you that later on again. So here we first get access to the BigBrain space, which is also registered in Zebra, and then we can, for the space, retrieve the template, get template, and this is exactly as with the parcellation map. That is an object that still does not include the image data. It only describes where we can find it. And if we want to see the BigBrain, we can say fetch, and we get a nifty object. Now you might ask, if the BigBrain is 1 terabyte in size, how does that work? I cannot have a nifty object here that is 1 terabyte in size easily. What Zebra is doing here, it will fetch it at a lower resolution automatically. Basically, we probably see even the output here, I'm not sure. Oh, it has been suppressed. Is it here? It's not shown here. But what it will actually say is it exceeds a feasible download size, so I'll fetch it at lower resolution, and then you can still choose to specify a higher one. Now we get it at lower resolution, but we have the whole brain as a nifty object. We can use again the nylearn plotting module to view it, and you see here there's the BigBrain at a reduced resolution in 3D for us to work with in Python. When we want to go to the full resolution, what you typically do is, in the fetch call, you can specify a resolution in millimeters. 0.02 would be the full resolution of 20 micron. You could also say resolution millimeter -1. This denotes the largest resolution anyway. But then you would really download to 1 terabyte. Zebra would refuse if you don't force it, and I would not recommend it. So if you want to look at the higher resolution, you typically specify a bounding box. Yeah? And specifying a bounding box is very easy. Zebra has a possibility for this, so you can simply say here, I want to have a bounding box that is spanned between 2 points, and you have to specify a space, since Zebra deals with different template spaces. So any spatial object that you generate in Zebra will always be tagged with a space. Zebra will not allow you to specify a spatial object without telling you which space it is. So we have to say here it's in the BigBrain space, and we span here just a bounding box between 2 arbitrary coordinates. That's of course hard to do because these coordinates will not be very meaningful for you. Usually you will have these coordinates coming from somewhere else. This could, for example, be a peak in your fMRI data, or it could be a position in the online viewer where you say, okay, I take this position with me to my Python code. Then you can just copy it and put it in here. Or you can use the annotation that we just made. You might have remembered that we copied this JSON representation of the annotation, and in siibra-python you can generate an object from this annotation. I will show you in a second how that works. So usually when you do such things, you bring in a location from your context, and with that location, when you go to high resolution, you typically specify volumes of interest, and these volumes of interest can be passed over to the fetch command, and then you can actually fetch high resolutions. You still get a nifty image, but now you have a chunk of the BigBrain at full resolution, which has still a feasible size. And the nice thing is that Zebra correctly tracks the space. So this nifty object, this nifty image, has the proper spatial information there. It still knows where it sits in the BigBrain space. That is why we can easily overlay this high resolution 20 micron chunk with the much lower resolution whole brain nifty image that we have downloaded here. We can simply superimpose them and they will sit at the right place. I think that's done here, is it? It is, but I can here, of course, here in the documentation I cannot zoom in. But actually, I'm not sure if you see that. Actually, here's a little orange. Even zooming in doesn't help. So if you do that in your own code, it's actually here. Yeah. So the 20 micron chunk sits here at the right position in the BigBrain space. We will see that in more detail as we go into the notebooks now. Yeah. Okay. I don't want to go too far here with looking at the example codes. Just one last thing. There's also a section here on the multimodal data features, and I use only one before we go into our tutorial notebook. This shows how you get the multimodal data features. So what we do here is we want to access reset neurotransmitter densities for a particular brain region. So we get access to a parcellation. We use the Jülich parcellation. Here's an older version actually. Let me go a bit further below. That's not what I want to show you. And what we can then do is we can ask Zebra to give us a region. This would be the primary visual cortex of the Jülich brain. This gives us a region object, and we can then query features with that region object of a particular modality, and this modality here is the receptor density fingerprints. Right? These modalities here they autocomplete. So you can just if you're in the Python interpreter, you can type zebra.features and tap, and it will actually show you what's there. You don't need to know these names by heart. You can really browse them as you do the program. So at that stage, you get a data feature, and these features typically allow you to plot themselves and so on. And what you will see is the same data that we also saw in the viewer, right? On the Python side, as as I showed on my slides, once you really request the actual data items, this will typically be interoperable data structures. We have already seen that the image data typically comes as a nifty image object. Such tabular data comes as a pandas data frame. Yeah? So once you did this call here, you have a pandas data frame, and you can do all sorts of stuff that you are used to do with pandas. All right. Again, a lot of talking. I will now go with you into the notebook that we have launched. So you should have a similar view that I have here. Those of you who are not familiar with Python, the notebook consists of cells which can have programming code snippets or text, and you can just run them by, I usually use Shift-Enter to go from cell to cell. You can also use the run button here on the top. So, and it doesn't work because we're working here with a new version. So you should actually first run this. So we released a super fresh version for this notebook, which is not yet, on EBRAINS is at least not yet there. Maybe on my binder it is. I have to reload, right? Now it's working, right? So now I have actually loaded Zebra. I also loaded some standard libraries. And again here the plotting library from Nylern, which we use a lot. So what we do in this notebook is actually similar somewhat to what we did in the viewer. So we will find a brain region. We will use that brain region to go to the BigBrain space and extract some high resolution data here. Yeah. So first thing is just specifying a brain region, and I do this here by keywords. So I just store a specification of the region. For P, that was the motor region, and a hemisphere, that's the right hemisphere, that's just a word, right? A keyword, nothing else. And then I get the Jülich brain parcellation, and from the parcellation, I ask for the region combining those two, right? So 4p, I just built the string here, 4p right. It's I split this because I will need the hemisphere later on in the code. Yeah, so I could just as well write here 4p right would work as well. Right? So let me just execute this. And if you look here now at type, what type that is, you see that Zebra has a particular type for the brain region, and it has a lot of attributes which we will not all use, but we could, for example, see if it has children and or parents, since this is in a region tree. So, for example, the parent would be the area 4p across both hemispheres, and so on. And the parent of this would be the precentral gyrus. So there's a lot of information already in that region object, but it's just more or less just metadata. What I then do is I get access to the map of the Jülich brain in the MNI space, and I choose the statistical maps or the probability maps. Yeah, and then I fetch the probability map of the chosen region from there, which then gives me a nifty image. This might take a little. When you use siibra-python, it builds up a cache on your machine. So when you use it often, it will have many things cached already. But here in the cloud service, of course, there is an empty cache. So whenever we load something, it will start loading things. Here, when I retrieve the map, it is really now loading the map from the online resource. But the second time I will use it, it will already have it available locally. Yeah. So I have now fetched the probability map. That is a nifty object. Let's have a look at that. Type pmap. You see that's a nifty image. It's not anymore Zebra object, as we explained. So I can just plot this with Nylern. That's the next cell. And you see here, we see the probability map here in this class brain view in the MNI space that works. You see how it is. So it's as we requested, it's in the right hemisphere, and this is where you would expect the motor cortex. Now, you remember in the viewer, we actually switched then to the BigBrain view when we were zoomed in the probability map of the motor cortex. We switched over to the BigBrain view and we saw the BigBrain sections that were at that location. We are now doing the same step here in Python. Of course, that doesn't work the exact same way. So what we will do here is we will we cast this nifty file into a volume, into a zebra volume, and then we can use this to run a query. Yeah. So as we did it in the code example with the neurotransmitter receptors, where we queried features by the region, we are now querying features here by the brain volume, by an image, but it's the same function. So we want to search features in the volume of that probability map, and we are interested now not in the receptor densities, but in the cell body stain sections. So this is a different modality. It's a different type of object we throw in to query the features, but it's otherwise the same mechanism. Then we get a list of sections, and from that list, I just take, since there will be several ones, I just take the one in the middle. So just arbitrary, yeah? I will just use one of those sections. So let me do that. This will also take a little bit. So it loads the instance. You see that there are 145 histological sections already available in the system. And now it is actually matching them. Oh, this also takes a little bit. It's now matching them against this brain region. This is also once you did this, these things will be much quicker the next time because Zebra is caching these information. But now it will actually look at each of these cell body stain sections, find out what their bounding box in the BigBrain space is, cache this information on your disk, and in the future it knows it. As I said, after that, Zebra will know the bounding boxes of all these sections. So when we use a different brain region and run the query again, it will be much, much faster because Zebra has stored the bounding boxes of these volumes. We're here. So on my side, it's done now. We have picked a section. Let me see what that is. Type section. That's a Zebra object, the cell body stain section, which is actually an image volume. So what we do now, in the viewer, we looked at the cortical layers. And here we have now a 2D image. So what we will do now is we will load the cortical layer parcellation, get the map in the BigBrain space of the cortical layer parcellation, and then I want to intersect the surfaces of the cortical layers with the image plane in order to see the contours of the cortical layer maps, right? I could also download the volumes with the segmentations, but here I really want to intersect the surfaces with this image plane. And the way we can do this now, you see that this is an experimental feature. We implemented this not too long ago. I can generate a plane with Zebra from the image section. Yeah. So then I get a 3D plane object that represents the plane that is coplanar with the image section. And then I can use this plane here to intersect each of the surface meshes from the cortical layer parcellation. And there are, of course, six of those surface meshes. Yeah? The gray-white meta boundary and the surfaces of so actually 7 surfaces of the different layers. And then, let me just do this. This intersection, you see already here, the plane for the section has a normal 0, -1, 0. So that shows us that the plane is actually in the Y, in the coronal plane. What we got now is we intersected all the surface meshes with the plane, and of course each surface can lead to multiple contours, because if you cut it with a plane, it might be that the surface mesh is interrupted. So what we get there are basically lines, polygons in the plane, and each surface might end up being split into multiple polygons. And since the BigBrain is not symmetric in the native histological space, you also see here differences, right? So some surfaces in the left hemisphere were split in more parts than in the right hemisphere. It's not symmetric. Here we just plot this. So we do the exact same things that you know already. We fetch a very low resolution version of the 1 micron histological section, actually at 0.8 millimeters, so very low resolution, the whole section, and we show the whole section. Let me run that already. Then we plot the section now with matplotlib, not with nylearn, which means we really plot the pixels only. We forget about the spatial anchoring and the affine matrices. That's why it also appears rotated here. But this allows us to also project the contours that we observed into the pixel space. Yeah, we can project them into the pixel space by using the affine matrix of the image, the inverse one. So the points of the contours, which are defined in the BigBrain physical space, are now projected into pixels, and then we can just plot them with matplotlib. And you see that here it's rotated because matplotlib does not know about the affine matrices. But you see now here we have a very low resolution version of the section. That's per se not interesting now, and we see the contours of the cortical layers on top of it, which have been intersected with that plane, right? If you look around, you might find some places where they're actually split. It's actually beautiful to look at. Thanks to Konrad Wachstil, who did this great data set. Okay. What we want to do now, it's a bit different how we approach it in the viewer, but actually I would be interested to extract a cortical patch at full resolution from the 1 micron section, which is correctly rotated and sits in the region that I selected above. So remember, we selected the region for the motor cortex in the right hemisphere. So I would somehow now like to find an automatic way of sampling a cortical patch from that region. And we will use both the region and the cortical layer maps to do so. The first thing we do is we find a contour point in the layer 4 surface, which is very likely to be in the motor cortex. How do we do that? We collect all the contour points of the layer 4 from the contours that we have just intersected with the plane. Yeah. So I just collect here all the points of the contour that are in layer 4. Create a point set in Zebra from that, and then I use the probability map. That's this line here. I use the probability map of the motor region that I fetched further above to evaluate those points. This basically gives me a list of the values in the probability map for each of those points. And then I just take the point with the maximum value. This is the point on the layer 4 surface in the tissue section that is most likely in the brain area. Yeah. And then I use this class cortical profile sampler to find a cortical profile in the cortical layer maps, which is closest to that point. Remember, the cortical profiles in the cortical layer maps, they are defined by the vertices on the surface. We intersected the surface with the image plane, so the vertices themselves, they might not sit on the image plane. They might be a bit away from the image plane. This sampler here finds the profile which is closest to the contour point that we identified for the motor cortex. So we do that now. This shouldn't take too long, I think. And you see it says basically the vertex 421,917 in cortical layer 4 on the right hemisphere is the vertex in the surface mesh that is closest to this highly probable point in the motor cortex in the section. And now we can use this profile. This is just a set of points. We can use this profile to actually create a cortical patch that is properly rotated, that sits in the image plane, that is properly rotated and is centered along the cortical profile, projected into the image plane. That sounds a bit complicated, but it's actually not that bad. So what we first do here is we fetch the part of the image section that refers to the brain region by intersecting it with the brain region volume. Then we fetch this at a resolution of 0.2 millimeters, so a bit higher resolution. We plot that just to see the image from a bit closer. Then we intersect all the contours with that zoomed in section view, and we show the cortical profile, the projection of the cortical profile that we have just identified, which should be a cortical profile in the sitting in the brain region. So here we are. Yeah. This is the 1 micron section at a resolution of 1/5 of a millimeter with the intersected boundaries of the cortical layer surfaces. And here we see the projection of the cortical profile that Siibra has found, which is close by. So actually this point here that we see here, that's the point on the layer 4 surface that had the highest probability. And this is the projection of the cortical profile that is closest to it. And we now use this cortical profile, so these points, to extract a patch at full resolution of 1 micrometer and rotate it that it's in the proper upright rotation. How do we do this? We have the object already that represents the image plane from above, and this object can actually define us a patch that encloses the cortical profile, so the set of points. So this gives us basically a canvas of the patch and the proper rotation of the patch. Maybe just forget about these lines, just say a few words about that. Since the image plane doesn't know that this is a cortical profile, it cannot know that the lower or the upper point of the profile has a specific meaning. So we know that the cortical profile, the first point, is the inner surface. Yeah, but the image plane just gets a bunch of points. It doesn't know that. The image plane just looks at that. It computes the principal component analysis to get the orientation, but it doesn't know that the first point is actually the bottom of the cortex or something. It cannot know. So what I'm actually doing here, and this can lead to a 100-degree rotation. It simply doesn't know whether its patch should be upside down or the other way around. So we use these lines here just to check that. So if it's upside down, we just flip it back. But that's not super important. So we get a canvas of the rotated patch, and we can use this canvas of the rotated patch to extract a subvolume of the brain section and properly rotate it. This will take a bit because here I'm actually extracting at 1 micron now. Yeah? This is a very small patch, so it's not too huge. It only fetches the 1 micron information in the local proximity, but this still will take a bit because 1 micron is just a lot. So while we are waiting for that, the following cells are all super simple. So after that, we will just display what we have. Yeah. So maybe while this patch is loading, oh, it's already there. Then let me run the rest. So now we have the patch. Here, I really just plot the patch with the NiLearn library. I do also fetch the BigBrain template at almost a millimeter resolution. And I plot the patch on top of the BigBrain template, just that you see that the patch still has the proper spatial metadata, right? So here we see the rotated patch at 1 millimeter resolution. So you see I can zoom in. I really have the cortical patch here extracted, which refers to this profile. Now let me show you the profile again. It was here. That's where we had the profile. And now we have here the corresponding patch. And you can see here in the next view, so this is just the same thing. I just plotted on top with the BigBrain, the lower resolution version of the BigBrain as a background image. So here, if I zoom in, that was a bit much. You can really see that the patch, it's a nifty image object, as everything is when it's image data. And you see here that it really sits at the proper position in the BigBrain space. It should. Yeah. But we have the full resolution. And here at the bottom, I just, to show that the patch has been resampled and sits upside down, I show the same patch, but I show the real pixel data with matplotlib. And here, so this is now not in the, this is in the pixel space, and you see that it's properly rotated upside down. I can even show the parts of the layer contours on top of it that we extracted above. And just for fun here, we are also plotting the name that siibra has produced automatically for this patch, and you see that siibra has tracked the information of the whole process. Yeah, so it says this is a rotated patch with corner points so and so in the BigBrain space, with image data sampled from section 3698 of this data set, and you will even be able to find from the Python type the DOI of the data set again, which I'm not doing here. Yeah. So at the end of this long notebook, what was the story here? The story was more or less the same that we did in the viewer. We selected a brain region just to specify the name. Then we got a brain region object from the Jülich brain atlas, and we fetched a nifty image with the probabilistic map of that brain region in M&I space. That's how we started at the top, right? Let me just go there. So we had a probabilistic map defined in the M&I space. Then we just used this map to find sections anchored to the BigBrain model which overlap with this map. Yeah, and this was just this little call features.get. What happened here in the background is that siibra translated the bounding boxes of the BigBrain sections into the M&I space in order to determine whether they intersect with the probabilistic maps, right? You didn't see that. It's happened in the background. Yeah? But it actually extracted from the BigBrain sections the bounding box. It knew they are in BigBrain space while the probabilistic map is in M&I space. So it automatically warped the bounding boxes into the M&I space to determine which ones overlap. This gave us a list of sections from which we have chosen just one randomly. And then we have used this selected section to define the corresponding plane in 3D in the BigBrain space. That happened here. And then we intersected the surfaces of the cortical layer parcellation with this plane, which gave us contours of the plane, contours of the cortical layer surfaces in the image plane. There, we actually looked at which points of those contours have a high probability to be exactly in the motor cortex. We used the probabilistic map again for this. Yeah? And there again, siibra automatically translated between the spaces because the probabilities are defined in M&I space. The contour points that we extracted for the layers are defined in BigBrain space. So siibra, we didn't really do that explicitly. In the background, siibra translated this because everything is tagged with the reference space and siibra knows it and it does it for us. So yeah, this actually happened here further below. Here, where we said evaluate points, right? The probability map was in M&I space, the points were in BigBrain space, and siibra still did it and just solved that in the background. We could actually always see that. So we have the PMap volume, and it has a space, and you see that and the space has a name. You see that this is the ICBM space, and then we had the points on the layer 4 surface, and they are BigBrain space. Yeah? And siibra uses this information in the background to translate properly. So it is really aware of spaces and deals properly with it. So then we found out which point on the contours of the layer surfaces is most likely in area 4P. And then we used this point to get a projection of a cortical profile at that location, yeah, in the image plane. And with this projection of the cortical profile, we use this to define the patch, because this gives us information how thick the cortex is, what the orientation of the cortex is at this position, where's the inner part, where's the outer part. And then we use this information to define a patch and extract the 1 micron information from the tissue section. So we now have sampled a 1 micron resolution cortical image patch in proper upright position, which is very likely originating from area 4P. All right. That is mostly what I wanted to show you. It would be easy for me to show you many more things, but maybe I'm stopping here. One thing that actually I didn't show you, I didn't get back to that JSON file. The BigBrain workshop 2023, so last year's workshop, that's this one. I'm not running it. I'm just showing you a single thing there. You remember that in the annotation mode in BigBrain, I copied the JSON representation of the structure that I annotated. You remember that? And I really forgot this. So this JSON representation, you could really just paste it in as a string. It's not the same one, but it's a similar one. I just paste it in. Of course, you can also save it as a JSON and load it. And then there is a function zebra from JSON, which can actually create a zebra point set from there. Yeah. So we didn't use that, but I told you there are usually several ways of bringing location into your Python code, and that is also one possibility. You annotate something, you export the JSON, and then you start your Python notebook from that JSON, right? So that would be a way to delineate the structure in the viewer, take it, and from there do a reproducible code. Thanks a lot for your attention. Many greetings to Montreal.